Hello there, welcome back to another chemistry webcast. In today's webcast we're going to continue our description of structure of proteins. In the previous webcast we focused first on secondary structure, which dealt with things like alpha helices and beta sheets and beta turns, and then we moved into tertiary structure, which described the overall folding pattern of a particular polypeptide chain. In this first portion of the webcast, we're actually going to talk about in between those two extremes, because it turns out that in between secondary and tertiary structure are some important repeated patterns that people have observed in a lot of biological systems that give rise to biochemists making good predictions about protein structure and function. So the first one of these guys is referred to as motifs, and all a motif is really is a short stretch of secondary structure that includes more than one alpha helix or beta sheet. But the idea is that it comes together and folds in a particular way that's been observed in lots and lots of different protein molecules. There's lots of examples like this. We're going to eventually talk a little bit about alpha keratin here in a sec. But the first one to focus in on is what's called the Helix-Turn-Helix Motif. And the reason I mention this one is because we've actually run across this in our conversation of transcription factors already to this point. So if we look at what a Helix-Turn-Helix Motif is made of, it is what it sounds like. It's made of a couple of alpha helices that are connected by a turn. But the reason I'm bothering to tell you about this is because this particular motif structure is one that shows up in lots and lots of DNA binding proteins. So to put it in other words, the reason why we should care to know about the existence of these things called motifs is because if we see a protein that has a stretch of amino acids that's giving rise to a helix-turn-helix type pattern, you can probably predict that it might be a DNA binding protein just by knowing that it has that particular stretch of folded structure. So again, structure gives rise to knowing something about function. But it doesn't stop here at logical sorts of names. We can take this out to ridiculous extremes. And just to show you how crazy biochemists can be when they start seeing these motifs over and over, it turns out that we have lots of silly names for these things, most of which I don't need to have you worry about. The only things you have to worry about are the few that I give you in the objectives, where I'm pretty sure helix-turn-helix is one, and alpha-beta-vero might be another. But coming back to the silliness, there's a particular protein that people observe that is a calcium binding protein that's important for calcium signaling that we'll actually learn about more in the second semester. But someone looked at this stretch of polypeptide right here, saw an alpha helix with the beta turn and beta strand and then another helix, and said, hey, that kind of looks like a hand grabbing onto a calcium ion. So it got the name EF hand. Gotta love biochemists. So motifs, just to summarize, motifs, more than just a single bit of secondary structure, less than an overall polypeptide. As a second example, and again, the point of this example is to really drive home this structure function idea. So there's a protein that we all have called alpha keratin. Some of you have heard of it. If you haven't, it's the structure that's found in hair, fingernails, porcupine quills, etc. Now, if you think about what the jobs of a lot of these structures are, is that it's important for the organism to have strength in these regions to be sort of a protective barrier, or to have certainly to have strong structural integrity. So if you look at the molecular level about how this structure is put together, it's actually made of a couple of alpha helical structures twisted together in this particular protein called alpha keratin. So why would that give rise to strength? Well, if you think about it, in addition to having all the individual strength of the alpha helix itself, where you have the interchain hydrogen bonding and all that good stuff, you can almost think about this in the same way you'd think about strands of twine coming together to make rope. If you have one strand of twine, that's not going to be particularly strong. However, if you can come up with a way of weaving those strands of twine together to rope, you know that rope is very hard to break and has a lot of strength by virtue of having these multiple layers of twine twisted together. It's the same sort of deal for this particular protein. By having these multiple windings of alpha helices, that increases the overall strength in the protein, and it's also what's responsible for allowing it to propagate or grow in a long particular direction, because alpha helices, as you know, naturally tend to have long, narrow, extended structures, like hair, like fingernails, and so on. So there really is a relationship here between structure and function. And if that wasn't good enough, it's also interesting to point out that a related protein to keratin, which is known as collagen, and many of you guys know what collagen is. Collagen is that strong connective tissue that connects bones, but it's interestingly made of a triple helix. So think of it as stronger rope. But again, the fundamental structural point's the same. At the molecular level helps to give rise to predicting the overall properties of the molecule in question. So this is a great example of that. And I do want you to know what alpha keratin structure are, but the theme that I really want you to get out of this is more the structure-function theme and knowing what motifs are and how they're different from secondary or tertiary structure per se. Okay, so the other part that's sort of related to this in between secondary and tertiary structure is a concept or a piece of vocabulary referred to as domains. So domains are actually really, really important. Defined domains are what are called compact regions in a protein. So in a protein that may be joined by a flexible segment of polypeptide chain. So I'm going to use my research protein as the example because I think it provides a nice way of thinking about this definition of a domain here. So when we look at a cartoon ribbon diagram of the crystal structure of Cu-A, if you kind of just look at it at a big picture, is made of two basic chunks that are separated by a fairly short linker region. Cu-A, as you guys may recall from class or just talking with me over the years, is a transcription factor protein. Okay, so it's a transcription factor protein that binds DNA, and it turns out that the DNA binding occurs in this upper portion of the domain, and ultimately the DNA binding occurs through interaction of a Helix-Turn-Helix Motif right here that's out of phase with DNA. But the deal here is that it's made of two domains, a DNA binding domain, which is one chunk of the protein, and that's separated by a small interlinker region that's connecting it to an effector binding domain. So for my protein, these two domains largely act independently one of each other, although the function of this one depends on what happens here. So in my research, what happens is when I take a molecule like carbon monoxide, which I should write the proper way, CO, carbon monoxide will bind here to these heme groups, and it turns out when carbon monoxide, the effector, binds in this region, it causes changes of the positioning in this linker region, which causes the Helix-Turn-Helix motif, which is a little hard to see on this portion, to flip up and around so that it can interact with the major groove of DNA. But the idea here is about domains, and again, the key point about domains is that they're large chunks of proteins that together have a characteristic fold that relate to or are connected to other domains of the protein. So Cu-A is an example of a bacterial protein, but it turns out that this domain idea is really, really important in eukaryotic organisms as well. And what's really cool is that if you take a class from Dr. Waters, say, in genetics, analysis of a lot of genes has shown that the DNA coding regions, which we call exons, for individual domains of multi-domain proteins are usually separated from intervening non-coding regions known as introns. So the domain idea is so important in eukaryotic systems that a lot of times when you're putting proteins together in eukaryotic systems, you actually have coding region for each domain separately. And this is a topic we'll get into more when we get into DNA-related issues moving forward in the course. So domains are also, like motifs, are somewhere in between that limit of secondary and tertiary structure. This is not a whole polypeptide, it's part of one, but it has a particular folding pattern associated with it. All right. Oh, and then I suppose it's worth making one more quick wrap-up point, which is that when we think about the amino acid composition of a particular domain, so again we have our effector binding domain here, it's amusing to note that the amino acids that make up a particular domain in one protein do not necessarily have to be the same from one protein to the next. That is to say, you don't necessarily have to have a lot of conservation of particular amino acids, but but usually the kinds of amino acids from domain in protein 1 to domain of a homologous protein in protein 2 are going to be the same. So that'll be something we get into and we start talking about molecular evolution.